Well, hello everyone. Welcome to another NAMA webinar. I am here tonight with Dr. Alden Dirks. Say hi to us, please.
Hi, everybody. Thank you for being here tonight.
All right. We have a pretty good crew on board, ready to watch you tell us all about false morels, yeah, or whatever you want to call them. Now, I'm going to do a quick introduction and then turn it over to you. So I don't want to take up too much time on the intro. Dr. Dirks has their doctorate from the University of Michigan and now currently lives in Pennsylvania. And Dr. Dirks is a total expert in the whole false morel area, whatever again you want to call it, Gyromitra, Lorchel, which is, by the way, it's kind of new to me. I don't really call it that. And they are, I guess, an ambassador to kingdom fungi in general. So I'm really looking forward to your presentation. I'm going to sit back and watch. I'll be manning the chat and helping anybody through there that needs to have any questions. And Dr. Dirks, if you could watch the Q&A as we go, I would appreciate
Sounds good. Thanks so
it.
much,
Thank
Trent.
you.
Yeah, and thank you everyone for being here. I'm gonna see if I can decrease the size of the Q&A. I'll put it over here in the corner. As Trent said, my name is Alden Dirks, and I finished up my PhD last year from the University of Michigan. And my PhD was on these mushrooms that we called false morels, lorchels. We'll get into this a little later. Mushrooms in this family, Discinaceae. So this talk is meant as something of a review. I want to share with you everything I think you need to know to engage with these mushrooms, kind of a lot of background and exciting new results that we have generated over the past 5, 6 years. So let's get started. When people think about these mushrooms deemed false morels, they typically think about them in contrast to morels, to true morels in the genus Morchella. And these are highly sought wild foraged mushrooms that people love to eat and pay a lot of money for. In field guides and culturally, people typically talk about these false morels as toxic lookalikes. They contain this mycotoxin called gyromitrin, and you should avoid them at all cost. However, it's much more complex than that, and there's a lot of diversity here. And as an ambassador to Kingdom Fungi, I've been doing a rebranding campaign. So I like to call these mushrooms Lorchels. I'm trying to give them a name that's not what they are not, but their own thing. And Lorchel is a term, a common name that's been used for these mushrooms over the years. So Lorchels are really fascinating, interesting, and some of them are quite toxic. However, paradoxically, they're also consumed as a delicacy in some parts of the world. So here we have a video. Hope that wasn't too loud for you like it was for me. We're showing here in Finland how these mushrooms are prepared for eating. They're detoxified by boiling them for 5 minutes and then rinsing them, repeating that process, boiling and rinsing them again. And then they can be chopped up and cooked up, sauteed with onions, with cream, and they're rendered safe to eat. So particularly in Scandinavia, these are really popular spring mushrooms that people enjoy eating. And this video really makes me want to eat some of these right now. So here they're making a little omelet with Gyromitra esculenta. Yummy. So this paradox of toxicity but delicacy has resulted in these lorchal mushrooms having a lot of lore, and people get really passionate about them. But we have to remember that gyromitrin is indeed a potent mycotoxin. And if one is unfortunate enough to be exposed to gyromitrin, symptom onset typically occurs 6 to 12 hours after exposure. Common and mild symptoms are vomiting, abdominal pain, and diarrhea. Severe symptoms include neurological symptoms like shaking and confusion that can progress to seizure, hepatic coma or death of the liver, and death. In rare cases, people can die as well from too much exposure to gyromitrin. There's also some interesting literature on the long-term effects of gyromitrin. It is genotoxic, so it can result in cancer. And there's been some interesting studies coming out from 2021 in the Alps. There's a cluster of people with neurodegenerative diseases that have been linked to their consumption of Lorchel mushrooms, resulting in ALS-like symptoms. And sorry, and someone asked, can geography affect toxicity? So yeah, we're not quite sure. These toxic mushrooms seem to be toxic wherever they are in the world. They're quite toxic. These are the only chemical formulas I'll be showing for you today. We're not going to get too much into gyromitrin and its chemistry, but just so you understand how these mushrooms can be rendered safe to eat, gyromitrin is this small molecule. It's water soluble, it's volatile, and it's acid and heat labile. That means it's broken down or it's changed in the presence of acid and heat. Gyromitrin corresponds to this first substance here, acetaldehyde, N-methyl, N-formyl hydrazone, but it has these 8 higher aldehyde homologs that exist in smaller proportions. So when you take Gyromitra mushrooms and you put them in water and you prepare them, you're taking these gyromitrin and homologs and you're breaking them down into N-methyl, N-formyl hydrazine, ultimately resulting in monomethylhydrazine. Monomethylhydrazine is an industrial chemical that's also used as a rocket propellant. It's highly toxic and carcinogenic, and it's the only mycotoxin known to poison you via inhalation. So the same reason how you can prepare these mushrooms to render them safe to eat also makes them dangerous because you can get sick by breathing in the fumes. So this is some background on gyromitrin. We're actually not going to be talking so much about gyromitrin in this talk, but I just want to give you that background as an important part of Lorchel lore. Because there has been a lot of disagreement and arguments over the years around gyromitrin. And part of that confusion comes from the fact that this group is actually a really diverse family. Gyromitra, the species we saw, belong to this family of diverse mushrooms, and there's been a lot of misunderstanding around their species diversity and species concepts. That's because there's a lot of old taxa in this group, a lot of species that were described in the 1800s with 1 or 2 sentences, and no pictures, no drawings. So we have a lot of old, inadequate species descriptions. Mushrooms in this group are frequently misidentified. When we start going into the fungaria and we are sequencing DNA barcoding specimens, they're very often misidentified. There's unclear species boundaries. We have inconsistent species concepts in the group as a result. And finally, the reports that do exist about poisonings and the chemistry studies that have been done, we can't really verify them because we actually don't know what species they were working with. The past 5 or 6 years or so, myself and colleagues, we've been using some modern molecular phylogenetic tools to try to make sense of this confusing taxonomy in Discinaceae. And I'm sure people who have been attending this talk, you've probably seen a number of these phylogenetic trees. So using either single markers or loci or a few loci, typically ribosomal DNA and maybe a few protein coding genes, molecular phylogenetics has just really revolutionized mycology. We've learned so much about fungal evolution. For example, crust fungi, another group that I'm really interested in, they used to all be clumped together in one family, Corticiaceae. But now we know that crusts have evolved 96 times. And that's because when we look at genetic data, we're actually peering more closely into evolution because evolution is operating fundamentally at the genetic level. And we're seeing that fungi are much more adaptable and there's a lot more convergent evolution than we ever imagined within fungi. So using these tools, we're making sense of this diversity, we're identifying species precisely, and we're making sense of things that have confused mycologists for a very long time. And since we'll be seeing some of these phylogenetic trees, I just want to give you a little bit of an overview of how to read these. So as a reminder, a phylogenetic tree, in this case, we're reading it left to right. And as we move to the right, we have more time or more genetic change. All of the names, these leaves listed here are species or taxa or whatever. And these are connected with branches going back to nodes. And things that share a more recent, are connected more closely by a node, share a more recent common ancestor. So using, for example, ITS, internal transcribed spacer, ribosomal DNA, we've learned a ton about species relationships. However, these one or a few genes can sometimes struggle to infer the evolutionary history deep in time. And it can result in poor support for branches and for nodes, and it can result in situations here like polytomies where we don't actually understand the branching relationship. Recently, there's been a transition, increasingly so, to using phylogenomic data. Phylogenomics takes hundreds to thousands of conserved genes, and it infers an evolutionary tree based on the combined patterns and history being told by all those different genes. Here is an illustration with this software called Discineae showing all the different patterns of all the different genes. And what we see is when we start actually looking into the genomes of organisms, it's a highly complex place and you get sometimes conflicting stories. That's because genes don't always exactly follow the same evolutionary trajectory because of things like horizontal gene transfer and incomplete lineage sorting. However, phylogenomics is our most powerful tool right now to make sense and infer evolutionary history deep in time. And that's what I did, and I'm going to share with you in this work, our phylogenomics study of Discinaceae. So using these modern molecular tools, we've addressed some fundamental questions in Discinaceae that I'm going to present to you here in this presentation, including what is the evolutionary history and taxonomy of the Discinaceae family? And what are their trophic modes or ecological guilds? Basically, how are they getting their nutrition in the environment? What are they eating? Which species produce gyromitrin and how did it evolve? How many species are there and where are they found? And how can you participate in the documentation of Discinaceae fungi? So now we're going to move into the section on our phylogenomic study. And this was published recently in Molecular Phylogenetics and Evolution. And any of the studies that I show throughout, most of them should be available. All of them, if they're my studies, are available on my website, so you should be able to access them free. But if you ever need help accessing any of these, you can just email me as well. And if you're interested in more of the methods and background on this, please look at our study. I'm not going to go into those right now. We're just going to jump right into our results. So here's our phylogenomic tree for the Lorchel family Discinaceae. And this has 62 Lorchel genomes that we sequenced. It was inferred with over 1,500 Busco genes. These are single copy universal benchmarking ortholog genes. Two different approaches to make these tree concatenation and coalescence approaches. They resulted in identical topology. And Bootstrap, which is commonly used to show support for phylogenies, was 100%, which is great. So what I'm showing you here is something called gene concordance factors. And basically, this is showing you what percent of those 1,500 gene trees support that particular node. So this one here I'm showing you right here is 62. That means that 62% of those 1,500 genes showed that particular branch. So the first thing I'll note here is that this top clade corresponds to our Lorchel family Discinaceae. These other groups here are outgroups. So we have the Morels, Morchelaceae in mint green, and we have the truffles, Tuberaceae down here. And then we'll talk about this yellow group in a moment. So this phylogenomic tree resolved the early evolutionary history of Lorchels. Previous studies using single or a few loci, they could never fully resolve the relationships of all these major clades. But using this much larger data set with pretty extensive sampling, we're able to confidently resolve the evolutionary history of Discinaceae. As I said before, all branches had 100% bootstrap support, which is great. Looking a little more closely at gene concordance, we found that this was generally high, and as phylogenomics go, again, genomes have lots of genes, and they actually typically have very complicated stories, and it's hard sometimes to find a full resolution. But here we found that most nodes in this tree were supported by a majority of the gene trees, which is very good in phylogenomics. However, I highlighted here a few nodes that still had less than ideal support, the lowest one being 24%, which means only a quarter of the gene trees supported that particular relationship. This was still the best topology given the data, but it just means that other factors, biological factors like incomplete lineage sorting, means that even our most advanced tools to understand evolution are still having a difficult time for some of these nodes because of the complicated processes of biology and evolution. But all that's to say is that this is still a very high quality phylogenomic tree and allows us to do some important things. For example, we revised the Lorchel taxonomy given this increased understanding of the evolutionary history. We recognize two tribes in the group. These are tribes Discineae and tribe Gyromitreae, and 10 different genera. And we're going to go through all these different groups later on in this talk, so you can gain more familiarity with these genera and species, particularly the ones that might be local to you. What was really good and exciting is that these genera are all distinguishable by morphology. Through a combination of gross morphology and ascospore morphology, they can all be readily distinguished. And more on that later. We also had some interesting taxonomic discoveries. Some species that we thought belonged to Discinaceae, they actually formed a well-supported group constituting a new family. This is entirely new, no-name family. And these species are Marcelina donadineae, collected from Spain. And then we have this other cup fungus, Paradisina melaleuca, from France. And this group definitely requires more follow-up research because we don't even have a name for it right now. And someone asked, are Halvella completely unrelated? Halvella are not completely unrelated. Halvella are, if I remember this correctly, where do they branch? They're more closely related to the Tuberaceae, I believe. Halvellaceae is not represented on this tree because there are no genomes for Halvella species, which was a bit surprising to me. And it's outside of the scope of the work I was doing. But yeah, Halvella really needs, someone needs to do what I did here on Halvella because there are no genomes for that group. But that's a good question. And I should note that most of these Discinaceae species used to be classified as Halvella. So there are some similarities there. We also see that morphology is all over the place. Mushroom macromorphology, as we see with other groups of fungi, is a dynamic trait with lots of convergence. Here we can see in tribe Discineae, it's predominantly cup fungi. In fact, if you find a cup fungus that's Discinaceae, it belongs to tribe Discineae. However, you have two different independent evolutions of stipitate fungi within that tribe. If we look here down in tribe Gyromitreae, we have predominantly stipitate fungi, but then we have the truffles that evolved from most likely a stipitate ancestor. And this goes to show that just because mushrooms look similar doesn't mean they're closely related, and that we're seeing that again and again with different gross morphologies. And someone asked, what about Disciotis venosa? That is a Morchelaceae species. So we're seeing that down here in Morchelaceae. Disciotis venosa, as you can see, Morchelaceae also has cup fungi, truffles, and stipitate fungi. This new family that needs a name only has cup fungi, as far as we know so far. In Tuberaceae, people are familiar with the truffles tubers, but it also includes cup fungi as well. So we have a lot of evolution of different gross morphologies within these groups. Great, so we have this really good Lorchel phylogenetic tree. Let's look at Gyromitrin and ecological guilds. And the data I'm going to show you next has been generated and presented in these two studies. A study from 2022, 2023, where we did a survey of the Lorchel family using this new chromatography, ultra high performance liquid chromatography method. And we documented and surveyed a bunch of different species to get a sense of which species have gyromitrin. And a follow-up study to that, to what I reported earlier, when people looked at this cluster of ALS patients in France, we actually tested those specimens for gyromitrin. So those two studies inform the data we're about to see here. Here we have our phylogenomic tree again, but here we have it annotated according to the presence and absence of Gyromitrin. So taxa that are in blue, that are highlighted in blue, were found to be Gyromitrin negative or not detected, and those that are in red contain Gyromitrin. Don't worry about the colors here yet, but I will point out that this is a time-calibrated tree, so this goes back to 80 million years, and what this is showing us is that the family Discinaceae has evolved and radiated into these 10 different genera over 80 million years. What we found is that Gyromitrin actually has a discontinuous and limited distribution present in the mushrooms that we've always known to contain Gyromitrin, the Gyromitra species, as well as surprisingly these cup fungi in the genus Pezizales. So as I said, it has a limited and discontinuous distribution, and this is actually suggestive of horizontal gene transfer. And we see this actually time and time again with secondary metabolites, including these things mycotoxins like amatoxins and psilocybin. We're seeing that secondary metabolites have a high propensity for horizontal gene transfer, perhaps because they're very useful, ecologically useful for organisms to have. We don't know the direction of this horizontal gene transfer. So the genes that make gyromitrin and the particular evolutionary history that resulted in this distribution are still open questions that I hope we can resolve in the future. And someone's asking, is there a rapid test for gyromitrin? No, there's not. It's not too hard, the test, but it does require a chemistry lab with expensive equipment that any chemistry lab has, but it does mean that you basically need to have a chemist on your team to be able to do these analyses. And that's unfortunately a pretty limiting factor right now. Just few people are willing to do this test, even though it's very basic, it's very simple. It just requires time and money that most people are not interested. And someone was also asking, is it difficult to sequence ITS from Gyromitra Discineae? No, it's not. It's not like Calvella. It's pretty easy, I would say, or straightforward. We've been able to sequence ITS from specimens from the 1800s, early 1900s. We get really high success rate even with very old specimens. And this distribution is suggestive of horizontal gene transfer, but that's because even though it's possible, it is less likely the alternative scenario where we can imagine we have a single gain event where an ancestral population of Discinaceae evolved the ability to produce gyromitrin. And for it to have this distribution, it would have to have been lost 5 times. So that's possible, but it seems less likely than a scenario of horizontal gene transfer. And horizontal gene transfer is this pretty wild, somewhat sci-fi concept that mycologists are starting to appreciate more and more as a pretty powerful force of evolution. It means instead of inheriting genes from your parents, from your ancestors, you're actually getting genes from distantly related organisms. And bacteria are very good at this. If we think about antibiotic resistance, that's because bacteria many of them are very good at transferring genes, resistance genes. But we're also starting to appreciate that that is actually a major force in the evolution of the chemodiversity of fungi as well. And Gyromitra gigas, what I'm calling actually Malblancomyces gigas, was analyzed. Here we have 2 specimens of that in this particular tree. And I should say that there are many more specimens that were analyzed for Gyromitrin that are not in this tree, because this is just the ones who had their genome sequenced. But Malblancomyces gigas, we have not detected Gyromitrin in that group. Here we're turning now to looking at the ecology of Discinaceae fungi. So we're going to start here on the left with the colors of these branches, which are showing change in the amount of one particular enzyme called AA9, the number of those genes in these organisms. And more on that later. Over here we have the size of the genomes in megabase pairs. Then we have the GC content. As we know, DNA has ACTG, so GC is just the percent that is GC versus AT, and 50% would be equal number of GC nucleotides to AT nucleotides. Then we have genes, number of genes in the genomes. size by the number of transposons. These are selfish genetic elements that jump around in the genome. Next, we have different cazyme classes, carbohydrate active enzymes, and then finally biosynthetic gene clusters. So this is a lot of data, but I'll walk you through it here. What we know with mycorrhizal fungi, so fungi that are forming symbiotic associations with the roots of plants, you typically find that they have an increase in genome size and a proliferation of these transposable elements, perhaps as an adaptation to this new way of life. Simultaneously, you have a decrease in these carbohydrate degrading enzymes called cazymes. And this makes sense because if you're getting your sugars and your nutrients from a tree, you no longer need to be breaking down wood and other organic compounds from the environment the way you would as a saprotrophic fungus, as one that's breaking down dead organic matter. And that's what we see here. The truffaloid taxa in the genus Hydnotria, they have a mycorrhizal genomic signature. They have enlarged genomes, as we can see here, with more transposable elements and a reduced cazyme content. We can see the reduced cazyme content in this AA9 in the colors of these branches. There's a severe contraction in the cellulose degrading enzymes, resulting in almost none of these enzymes to degrade cellulose in the Hydnotria truffles, whereas all the other taxa appear to have a saprotrophic genomic signature. Just kind of pretty standard size genomes, pretty standard repertoire of cazymes. So it's not a surprise that the Hydnotria truffles are mycorrhizal. We knew this already. I would say what's more of a surprise is that it doesn't look like anything else in this family is mycorrhizal. They all appear to be saprotrophic. And then someone was asking about the background rates of horizontal gene transfer. So this is all hypothesis-based right now, and I don't know the background rate of horizontal gene transfer. I think that's a difficult thing to estimate at a large scale. So interesting question though, but I think once we identify the genes that make gyromitrin, we'll be able to start actually testing some of these hypotheses, which will be really exciting. Great. And even though everything here has what I would say is genomic properties of saprotrophy, there are a few nuances that these genomic level metrics do not really capture. And what we find is that this bottom clade here, tribe Gyromitreae, includes species. Everything here we've been able to culture from spores. And they've also been isolated as endophytes, perhaps, from plants, especially mosses, as well as lichens. So endophytic and endolichenic fungi, also culturable from spores, this bottom clade. Interestingly, the mycorrhizal truffles we've also been able to culture, even though they're mycorrhizal. However, everything from this top clade, tribe Discineae, none of the spores have been able to germinate on standard media, and none of them have been isolated from plants or lichen tissue. So pretty interesting pattern we're seeing here that I think speaks to something deeper and fundamental about their ecology that is definitely going to require more follow-up research to figure out what's going on with these perhaps endophytic members. How come this other clade tribe Discineae, how come they can't grow in culture on standard media? Seems like there could be an ecological signature there. But what is interesting is this pattern here supports what's called the waiting room hypothesis. And this is a hypothesis for the evolution of mycorrhizal fungi, which says that fungi that are already associating symbiotically with plants are primed to evolve to be mycorrhizal. And we see that here with this bottom clade, tribe Gyromitrae, all these other taxa being endophytic in nature, we could say, it suggests that the ancestor perhaps was primed to be able to form this very intimate symbiotic relationship as a mycorrhizal fungus, resulting in the evolution of these mycorrhizal truffles. And someone was asking about gyromitrin being more predominant in West Coast versus Eastern specimens, and there's no evidence for this. I have found high levels of gyromitrin in both places. Why assume that is all or nothing with mycorrhizal or saprotrophic? So I think here I'm speaking to the complexity of the saprotrophy, whereas the mycorrhizal signature is quite clear just based on the data that's been accumulated over time. But true, yeah, that's a good point though, that it's not all or nothing for the saprotrophy. It does seem like there is more complication to the ecology there. So exciting to see how what we'll learn over time about the ecology of these organisms. So in summary, we used phylogenomics to infer a robust evolutionary history for the Discinaceae family, and we revised the taxonomy accordingly. We found that Gyromitrin has a limited and discontinuous distribution, suggestive of horizontal gene transfer, and Hypnotria appears to be the only mycorrhizal genus in the family. And with that, since I think one of the major changes or results of this study were the taxonomic revisions we made, I'm going to go into that component for the next portion of this talk. So here we're going to get into the taxonomy and biodiversity of Discinaceae fungi. And in this section, I'm hoping to introduce you to perhaps some fungi that are familiar to you already, maybe some new names they have, or introduce you to some species that you didn't know existed in your region and get you excited about those. And someone was asking about, can you culture these fungi whose spores do not germinate on standard media? Can you use tissue cloning? We haven't done so much of that, but my attempts at that also failed. But that is something that I would encourage if there's if there's anyone who's doing culturing work, give that a shot. At some point, someone thought they had done that with Malblancomyces montanus. It turned out to be a contaminant. So we have not had success yet with that, but that would be very cool to get cultures of these tribe Discineae fungi from tissue isolates. Okay, let's jump into our tribe Discineae, and we have our 5 genera: Discina, Malblancomyces, Neogyromitra, Pezizales, and Pseudodiscina. I assumed, and it looked like from the chat, that most of the audience is based in the United States, and this talk is geared to that audience. We'll be talking to you about species that occur in the United States predominantly. First up, we have our genus Discina. And for every one of these genera, I will be also telling you or showing you the traits that can distinguish them, particularly the spore shapes. So here we have Discina, and Discina is readily identifiable by these pointed protrusions at the end. We call these apiculi. They have pointy apiculi at the ends of the spores, and they have these 1 to 3 of these bubbles, which we call gutules or lipid droplets inside the spores. And finally, they have this ornamentation. It's can be like warty but usually forming a reticulum or this net-like pattern. So Discina, readily identifiable by these pointy apiculi. Discina is actually a potentially big genus. So far, we know it has at least 15 species. 3 of them occur in North America: Discina ancilis, McKnightii, and Warnii, which I'll talk about. In addition, though, there are 5 unknown species, 5 additional documented unknown species in the Pacific Northwest. Within this group, we definitely need to study some type specimens to determine the proper application of Discina ancilis, perlata, and Warnii, and other taxa. These are really old names, and it's difficult to know which name corresponds to which phylogenetic group. In addition, beyond the ones I mentioned here, there is many species that are actually described centuries ago in the 1800s, for example, with little morphological information and no sequence data, and they may or may not belong to Discinaceae. A bunch of Discina species that are kind of a catch-all. So a lot of old names that we'll have to look into, and perhaps they correspond to these unknown species. So here we have Discina ancilis and Discina warnii. These are both found in the eastern North America. And this is really cool because both of these specimens were found by Dave Wazalewski in Pennsylvania at the same location on the same day. They are very similar fungi. Warnii has somewhat smaller spores. but their ITS is only about 87% similar. So they're highly divergent. In Western North America, we have Discineae, and this appears to be the most common of these Discineae cup fungi in Western North America. And then down here at the bottom, we have two examples of these unknown Discineae species found in the Pacific Northwest. And this brings up our first theme, which is that there seems to be a high Discinaceae biodiversity in Western North America. And it's pretty unclear to me why that's the case, but we get a lot of, not we, y'all, whoever is out there, y'all get a lot of species out there in Washington, Idaho, Oregon, down into California. It's quite amazing, and also Arizona as well. It's amazing the Discinaceae biodiversity, and there's a lot of unknown clades coming out of sequencing efforts from those states. Next up, we have Mycoblancomyces. This is a genus that produces both stipitate and discoid fungi. And these are characterized by this blunt or these knob-like apiculi on the ends of the spores. This genus does include discoid species, at least 7 of them. Again, we have high diversity in the Pacific Northwest and Western USA, where there's 5 species, 2 of which are unknown. I'm not going to talk more about these. However, if you ever do find discoid Discinaceae species, as I just mentioned, you can pretty readily identify them as Discineae or Mycoblancomyces based on the shape of their apiculi. Within the stipitate taxa, these are some of the most iconic Discinaceae species. And there are 5 that occur in North America, 3 that are named. These are Mycoblancomyces americana gigas, Corfii, and montanus. And these might be some of the most familiar species to people. Used to be called Gyromitra montana, for example, now Mycoblancomyces montanus. And these stipitate species have been the subject of 2 studies over the past 5 years or so, part of a series of studies being conducted on the taxonomy of Gyromitra and related species being led by Andy Miller and Andy Methin. And these studies have clarified quite well the species boundaries and concepts within the stipitate Mycoblancomyces. And since these studies were done before the change in name, they'll have the genus Gyromitra attached to them. So within North America, we have Mycoblancomyces americana gigas, Corfii, and Montana. If you're out in the West Coast, things are pretty simple for you. You just have Mycoblancomyces montanus. If you're out here on the eastern United States, things are a little more complicated. And we need actually more specimens to be sequenced to clarify the situation. We mostly have Mycoblancomyces corfii and americana gigas, but you can see here there's actually also Mycoblancomyces montanus documented in eastern North America. And when you find one of these, it's really hard to know which species you have. So you could have 1 of 3 species if you're collecting these out here in eastern North America. They're pretty much morphologically indistinguishable. However, they are all edible, and they're considered quite good as long as they're thoroughly cooked. And this brings up our second theme, that there's actually a lot of morphologically cryptic species in Discinaceae. We're finding that species that used to be lumped together as one thing, we're finding 3, 6, a dozen different species, all that could have morphologically been considered that one thing. And what I'm also showing you here is that Mycoblancomyces americana gigas, this is a species that I collected in Michigan that we described as a new species a few years ago. You can see here it's growing and almost has this discoid form. And it's cool because it shows you that transition between discoid and stipitate. And it doesn't take much. These stipitate fungi are basically cups on a stick. And if they don't have much of a well-developed stick, they're basically discoid fungi. And so it doesn't take much for these fungi to evolve into these different, what we call gross morphologies. As I mentioned before, there are 2 undescribed species in Alaska, and these have been discovered through some sequencing work of specimens from Alaska collected by Phyllis Kempton and Virginia Wells. Over 45 years, every summer, they collected mushrooms in Alaska. They were collaborators of Alex Smith and shared some of their specimens, and they published their own research as well. In 2001, 7,000 of their specimens were donated from Alaska to the University of Michigan. sequencing these, finding that actually, yeah, there's these two undescribed Malblancomyces species. And here is one of them. This is a specimen. They collected it. You can see all their notes from their specimen. They didn't have, I guess there weren't too many spores. They thought it was immature. They actually identified it as Gyromitra fastigiata, which people might be more familiar with as Gyromitra caroliniana, or Neo-Gyromitra caroliniana, we'll be calling it. But then later on, Sean Abbott, an expert in this group, identified it as Gyromitra gigas. Now, of course, that's Malblancomyces gigas. And this, with sequencing, we can see that it's actually a distinct species. So Alaska seems to have a lot of interesting stuff that, and not any kind of modern collections of these kinds of things. And someone was asking, are Eastern North American Malblancomyces montanus found in coniferous settings? I'm not sure where those ones were collected. I would have to go and find that information. Yeah, good question. Next up, we have Neogyromitra. And this genus can be identified by their ascospores, which contain a series of little apiculi at the end, kind of these like little prongs or projections. They have more of a reticulum ornamentation, just like the other ones, but multiple little apiculi coming off the end, which is cool and interesting and so strange because you have these discoid and stipitate species, but they all have these little prongs coming out. And these taxa were the subject of another one of these studies in our series. And so this is brand new work where we've clarified the species boundaries and concepts here, including the description of a new species, Neogyromitra japonica. So we have 3 discoid species in Neogyromitra. None of these are found in North America, so I won't talk about them more, except I will say that Neogyromitra japonica seems to have a high sequence diversity. And some of these were collected in areas that perhaps were exposed to radiation from atomic fallout. And Andy Miller conjectured that maybe some of this is mutation from exposure to that radiation. So we definitely need more collections of Neogyromitra japonica from Japan to understand if it's one highly diverse species or multiple species. Within the Stipitate group, we have 3 different taxa. 2 of them occur in North America, just in Eastern North America for the most part, which are Neogyromitra brunea and Neogyromitra caroliniana. Here's a map from our recent study, and you can see that Neogyromitra brunea has a more northerly distribution and caroliniana a more southerly distribution. And I said mostly Eastern because we have this one collection, I think from Utah or somewhere maybe by the mountains, and that was a pretty big surprise. But these are really amazing fungi. Gyromitra brunea has these themes, making it easily identifiable in most cases. Sometimes these themes aren't very, they're more fused than this, and then it gets a little confusing. But then we have Gyromitra caroliniana, which is also called the big red or beefsteak fungus is another name for this. And both of these fungi are exceptional edibles. These are some of people's favorite mushrooms to eat. And again, I will encourage anyone who eats these fungi, of course, you need to positively identify what you're eating, and you need to thoroughly cook these mushrooms to render them safe to eat. But there is a widespread culture of people consuming these mushrooms, and they are just as safe as any other mushroom that's regarded as edible. Next up, we have Pezizales. And these are some cup fungi that have these really cool spores with these concave depressions. These have been described as fishtail apiculi, resulting in the genus name Pezizales, meaning fish cup. There's at least 3 species, maybe 4, in this genus. Pezizales leucoxantha and persicula are the most well-documented ones. Pezizales persicula was a species that I described as a new species last year from the Pacific Northwest. There's also one species known to occur in China that does not have a name. And then there's Pezizales convoluta, which is an old name that may just refer to a mature Pezizales leucoxantha, not quite clear. Again, these Pezizales cup fungi do contain gyromitrin, not that I know of anyone trying to eat them. So here we see Pezizales leucosantha. So this is a yellow cup fungus that's found in Eastern North America as well as Europe. And then Pezizales persicula. Persicula means little peach. So this is the little peach fish cup fungus. And that's because it has more of this peachy coloration to it. This was a fungus that Michael Bew collected over many years, and he talked about it in his book from 2014, I believe it was, Ascomycete fungi of North America. He mentioned it as an unknown species. So at some point he sent me all of his collections, and indeed this was phylogenetically, morphologically distinct from Pezizales leucosantha. So we described that as a new species. Over here you can see Pezizales convoluta. This is the packet in the New York Botanical Garden. Unfortunately, this is from the 1920s, I believe. 1921 it was described. Unfortunately, you can see that it's destroyed. There's no material left. There's nothing at all. And this is unfortunately pretty common for these old mushrooms. There's just no mushroom left. And it leaves you with this interesting dilemma where the name is still applicable. We could kind of just ignore it, but um, yeah, we don't know what this is, and we could try to go to the type locale where this was collected and try to find something else that matches its brief description and see if it differs from these other taxa, but it leaves us in a tough situation that we'll have to try to resolve in the future. Finally, for tribe Discineae, we have Pseudodiscineae. These are cup fungi, and they're a little different from all the other cups in tribe Discineae. They have smooth spores. Sorry, they don't have smooth spores. They have non-apiculate spores. They don't have any of those projections at the end, but they have these warts on them, and they're bigutulate. They have 2 lipid droplets instead of 1 to 3. So this species, when it was described and discussed in the literature, it was considered kind of intermediate between these 2 major groups. There's only 2 species in this genus so far, Pseudodiscineae melileucoides and yunanensis, the second recently described from China. Melileucoides is a species found in Western North America. And it's pretty beautiful. It's quite, yeah, it's quite beautiful. And it's pretty easily identifiable by this thin, somewhat elongated central stipe. Compared to the other cup fungi I showed you, they have more of a robust stipe. And also, of course, if you have a microscope, readily identifiable by the spores they produce. So if you're out in Western North America, Pseudodiscineae melileucoides is something you should be able to find. Great, let's move on to tribe Gyromitreae, including genus Gyromitra, Hypnotria, Paragyromitra, Pseudorhizina, and Pseudoverpa. First up, we have our genus Gyromitra, the most famous and classic Discinaceae group. These have spores that are smooth or unornamented, and they have no apiculus or a little bit of a broadly rounded apiculus, and they're bigutulate. They have 2, they have bipolar gutules. The genus Gyromitra, they're all stipitate species, and they have at least 5 species. The 2 I'll talk about relevant for us in North America are Gyromitra splendida and venenata. However, there may also be 1 unknown species from Mexico, but we definitely need more collections from that region of the world. These, again, are toxic and potent Gyromitra producers. In the video I showed you at the beginning of this talk, you can effectively remove Gyromitrin to render these safe to consume. And I'm not interested in trying that, and I wouldn't encourage anyone else just for safety. But if that's something that you do want to do, definitely make sure you read up and follow very closely those protocols, because these have very large amounts of Gyromitrin in them. So you just definitely need to be safe if you attempt to eat these. There's a lot of old forgotten names that might correspond to similar taxa that are listed here. So it's really difficult, and we definitely need to try to find these mushrooms and see if we can get sequence data from them to understand the application of taxonomic names to the phylogenetic clades that we are finding. Here's some photos of these taxa. On the left, we have Gyromitra splendida, and on the right, we have Gyromitra venenata. So if you're out in the Western United States, Gyromitra splendida is by far your most common species of Gyromitra. And these mushrooms are quite variable. Sometimes they have more what you could say a typical morphology, this brownish cap that's kind of brain-like and folded with this finished stipe that has this furrow along the side. You can find some albino versions though, which are really cool and somewhat rare and kind of an open question of why the heck these things are albino. And then you also find some that are much darker, like these almost blackish ones. Gyromitra venenata it seems to tend to be a little more robust. However, there's so much variation in these, it's pretty difficult right now to know how to identify them in the field. And this brings up our third theme, which is that we see this Eastern-Western USA split in biogeography for Discinaceae. This is not too surprising. We see this for all kinds of fungi and plants and animals, but it's not a perfect split. And what I mean by that is even though Gyromitra splendida is the most common Gyromitra species in Western North America, you do also get Gyromitra venenata and the same situation. Um, so we need a lot more collections and to understand why these things have their distribution that they do, and how they overlap with each other. So someone's asking, what percent would you consider to be a large amount of gyromitrin? What percent? I would have to check my notes, but I think these are somewhere around 2 milligrams per kilogram of gyromitrin per dried basidiocarp. Um, And that's kind of like the high levels we're seeing with these Gyromitra mushrooms. So yeah, I can't do the math right now. But if you go to those studies that I mentioned before, any of the highest levels that we documented would definitely just Gyromitra mushrooms in general. I haven't seen any that I would call low levels of Gyromitrin. Even though I think it would be hard to say like what is safe for a human to be exposed to. I don't think anyone has that answer specifically, especially when there's factors like ALS, as we talked about. We really don't know how much ALS you would need to be exposed to how much gyromitrin you need to be exposed to for ALS or genotoxic effects to be a problem. Um, yeah, good question. Great. Let's now move on to our truffles, Hydnotria. You don't need spores to identify these. These are the only truffles in the group, but they do have interesting, cool spores that are pretty variable. They can be kind of cuboidal or rectangular, or more ellipsoidal with some ornamentation, or just more globose with these large nodules on them. Hydnotria, I can't give you too much information on that, and that's because there's a lot of species in this group, but we don't have a good sense of which phylogenetic clades correspond to which species name. There's at least 17 species and a number of unknown clades, and I'm sure there's a lot of undocumented biodiversity in this group. That's because not a lot of people are digging up truffles. The only types that have been sequenced belong to species described from China in the past decade. So it's really impossible to know what species we have in North America because a lot of these species concepts are polyphyletic. Hydnotria cerebriformis, Cubospora, Variiformis, and Tulasne are some of the names that you see thrown around, but nobody knows what clade specifically those correspond to. A lot of work needs to happen in this group. But what we are seeing is that people are finding really cool specimens, especially again on the West Coast, either by raking or popping out of the duff in the moss, or sometimes in the case with Heather Dawson with trained truffle hunting dogs. Apparently Heather Dawson's truffle dog's favorite truffle is Hydnotria, which is cool. So yeah, these don't even know what to call these. There's a lot of Hydnotrias out there and a lot of interesting biodiversity there. Next up, we have Gyromitra. Here's a beautiful photo by Christian Schwartz of these with some of them, their violet tinged stipe. Gyromitra, they're all stipitate species, and we have at least 6 species, and 3 of them are found in North America. Gyromitra infila and ambigua have a very wide distribution. I would say both of these have a global distribution, at least in temperate zones. But there is one unknown species from Western USA that only has a few documented or sequenced collections. and we don't have any good photos for them, from California and Arizona, I believe. So we definitely need more of that unknown species. Paragyromitra species have been reported as being toxic, but we've never detected Gyromitra in this group. It's possible that they are Gyromitrin producers and the specimens we looked at just weren't making it. That's hard for me to believe because every other specimen we've tested in Gyromitra and Pezizales has contained gyromitrin and detectable levels of it, even old specimens going back to the early 1900s. So it seems like if something makes gyromitrin, we can detect it. However, what I think is more likely is that assuming these are toxic based on credible reports, they are maybe just making a different toxin. And this this point is important to make, and that's because every species of fungus is making a dozen or more of these secondary metabolites, these specialized molecules, these small molecules that are used in their ecological interactions. Think things like penicillin and amatoxins and psilocybin. The chemodiversity of fungi is massive and so desperately underexplored. So if these were producing some undescribed toxin, that would be no surprise at all because they're absolutely producing a dozen other things that no one's ever characterized before. That's just something to keep in mind. Is it possible that Gyromitrin evaporates from harvested fruit bodies was a question. Yes, and I would say Gyromitrin is probably always evaporating to a degree. However, it was really useful for me when a chemist told me about other volatile substances. Think about when you work with something like basil, right? It has that beautiful smell, highly volatile. However, you can dry basil and it still has that smell. You can still crush it up and you can smell that basil smell. And then you get something really sensitive, like one of these expensive chemistry instruments, and they can detect that chemical even if there's very small amounts of it. So you can take Gyromitra mushrooms, you can dry them, you can put them in a fungarium for 100 years, and then you crush it up with our test, and you can still detect Gyromitrin because even though it's volatile and it's going to be losing some as it's drying, it always retains some of it. Yeah, a great question. Here's some photos. On the left, we have Paragyromitra infula, and on the right, Paragyromitra ambigua. Ambigua, as the name suggests, ambiguous. It was separated from Paragyromitra infula. However, they look pretty similar. And besides spores, Paragyromitra ambigua has somewhat larger spores. The main difference is supposed to be that the Paragyromitra ambigua has this kind of violish tint to its stipe. But you can see from these other sequenced and confirmed specimens of Paragyromitra ambigua, that's not a reliable characteristic. You get some that are much more similar to Paragyromitra infula. So these can be difficult to separate in the field. However, what this photo is showing up here of Paragyromitra infula, Damon used KOH and showed that there was this really strong KOH reaction. KOH is used in a lot of other mushrooms, but in the literature, there are no reports of KOH reactions for these fungi. So this was really cool and exciting to see that KOH turns this such a red color and opens up the possibility that chemical tests could be useful for distinguishing Discinaceae specimens in the field. So if you're out there collecting Discinaceae and you have some of these chemicals, definitely test it out. And then there's a question here, have there been any analysis of gyromitrin in properly cooked Gyromitra esculenta? There have been some studies from the 60s and 70s when gyromitrin was kind of a hot topic in Europe because people discovered the substance. It's something that people eat as a delicacy. So people did do those analyses to see how much of it was lost over time. And by properly cooking it, again, it has a small amount left, but a very small amount. How much is too much Gyromitrin? Nobody can answer that question. We're exposed to toxins all the time. And it seems like the people who are eating these regularly, if they're preparing them properly, it's not so much to cause concern. But yeah, we really don't know, honestly. Great, let's move on to our second to last genus, which is Pseudorhizina. And these are really beautiful, cool mushrooms. And they have spores that are smooth, no ornamentation, no apiculi. They're either spherical with a small gutule that's not centered, or bipolar gutules. Pseudorhizina has 2 species, maybe 4. Californica and Sphaerospora are the most well-characterized ones. It seems like there might be 2 species going under the name Pseudorhizina californica in the West, but we'll need more specimens to determine that. And then finally, Pseudorhizina discinoides. This was a species described by Imai in Japan in 1932. It has spores that are similar to Californica, but doesn't have the same coloration that's common to Californica. But you can also find Pseudorhizina californica without those bright colors. At the same time, there are no sequence specimens related to this group in Japan. I wouldn't be surprised if it's its own species, and there's probably more species in this group throughout the world. Here we have some photos, Pseudorhizina sphaerospora and californica. I purposely chose these photos because before I'm showing you this really beautiful, bright Pseudorhizina californica. You know, this is just a gorgeous specimen and kind of what is characteristic supposedly for Pseudorhizina californica, these like violet colorations. But you do get some specimens that don't have strong colorations, and then they become very difficult to separate. Pseudorhizina sphaerospora has a global distribution, whereas californica seems to be isolated to the west coast. If you have access to a microscope, you can separate these pretty easily by their spore shape. Sphaerospora, as the name suggests, has spherical spores. Californica has ellipsoidal spores. But they both can have these kind of reddish, slight reddish tints, if not a very violet colored stipe in the case of Californica. So someone was asking, should we not be using the term Gyromitra esculenta for North American species and instead use terms such as Gyromitra venenata splendida group? As we currently understand the taxonomy, Gyromitra esculenta does not occur in North America. It has not been documented. We have Gyromitra splendida and Gyromitra venenata. You know, if you want to use the generic taxonomy that I'm using in this talk with these 10 different genera, you can just call everything Gyromitra. You know, venenata, splendida, esculenta, they're all Gyromitra species. So you can leave it at Gyromitra spa if you want. Gyromitra venenata splendida group seems like a lot or maybe a little redundant. You could, if you're on the Eastern North America, I would call it venenata at this point. If you're Western, I would call it splendida, knowing that that might not be 100% accurate. So, but I encourage people to keep working with these mushrooms, and we definitely need to collect more. I'll talk a little more about that later, the way you can help with our understanding of this particular group, the Gyromitra genus, because we definitely need help figuring out how to identify these in the field. Great, let's move on to our last genus of the night, which is Pseudoverpa. And this has these ellipsoidal spores, bigutulate. Pseudoverpa, as the name suggests, is verpa-like. There's only one species. This was described from Cyprus in 2018 from a burn site, so a relatively new species. It has only been documented from Cyprus and from California and Oregon, 2 specimens. So there's three specimens known in the world. These Western North American specimens may constitute their own species. We need more collections. They're pretty similar in terms of genetic similarity, so we can't say right now. It's a really interesting mushroom. Again, verpa-like with this cylindrical stipe attached at the apex. Here is the second known collection that is quite distinct. It has this kind of bluish black coloration. Although I think it's a little more difficult to identify these than I would hope. Here on the right, we have 2 pictures of Gyromitra splendida from California, both confirmed with sequencing. And you can see that I find them to be pretty similar in terms of having a smooth, somewhat cylindrical stipe. The cap is not fully attached to the stipe. Even here you can see with Pseudoverpa anthracobia, we have some attachment of the cap to the stipe a little bit there. So I'm finding it hard to know what is Pseudoverpa versus Gyromitra all the time, but we definitely need more collections of this. There's only 3 specimens for the entire world, so there must be a lot more out there and a lot more interesting biodiversity in this group. Great. I hope that was not too much, not too boring. I hope that gave you some insights into some of the species that you might find in your area and some of the known unknowns to inspire you to go out and start looking for these Discinaceae fungi. Now I'm going to move into some information for you, for mycologists and community scientists, to hopefully help in the documentation of these Discinaceae fungi. So here's some tips and techniques I have for you. First of all, it's good to look for Discinaceae fungi, and you can find other interesting Pezizales around moss and on persistently wet, bare soil. Alkaline sites are especially interesting. If you know sites that are calcareous, that have more of a basic pH soil, you get some really interesting Pezizales fungi in those places. Here's an example of where you might find a Pezizales fungus. This is in New York. We have this pool here, a really beautiful place. And then you have some bare soil that's wet on the edge of this pool. These bare soil spots are excellent for finding Pezizales. And here we have Paragalactinia succosa with these really cool kind of warty spores. Another example is this tiny little cup fungus. This was found twice in California. And this is a completely unknown species belonging to that family, if you remember, including Marcellina Donadineae and Paradisina melaleuca, a family that we don't even know what to call it yet, a whole family that, and this, we don't know what species it is, we don't know what genus is, we don't know what family it is. But from sequence data, we know that it's very distinct from anything else documented and it belongs in that unknown family. And this was found growing within moss. So again, moss and bare soil, wet places are really great for finding Pezizales in general. My second tip for you is, if you can, to accompany your collections with macroscopic measurements and microscopy of mature, freely liberated ascospores. So that's very important that they're mature and freely liberated. You're not measuring them within the asci, but they've been released already from the asci. And I should note here, asci are the elongated cells that contain the spores in this group that we call the ascomycetes. As I talked about in the taxonomy section, a lot of these have apiculi on their spores, and it's important to measure also the apiculi when you're measuring the ascospores. So take the entire length and then also measure separately the apiculi. And when I say macroscopic measurements, I mean when people are collecting Gyromitra, they rarely take note of the length of the cap or the width of the cap, the length of the stipe, and just basic information like that is impossible to retrieve from specimens once they've been dried, and very difficult to get from photographs, if not impossible. So if people are making collections of Gyromitra, even though it seems like these are everywhere and there's so many of them and so many people are documenting them, making those precise measurements could be really helpful for us, along with DNA sequence data, to start to make sense of how we might be able to start identifying these in the field. Because as it stands, there seems to be a lot of variation, and rarely do we have the data linked with a DNA identification to be able to make sense of these differences. And a question here, are there the saprotrophic Discineaceae, are they wood rotters? I don't know. I don't know if they are wood rotters or white rot, brown rot, or if they're eating, they could be eating soil, organic matter in the soil. You do find some Discinaceae fungi like Paragyromitra infila, as well as Pseudorhizina, growing what seems to be directly from wood. So yeah, that's a good question. Not sure what exactly they're eating. That will definitely be something we need to figure out in the future. So here, to the point of our measurements, I'm linking, or I'm showing you here a publication recently from Alison and Gregory Benito's lab at Michigan State University, where they nicely document how you can effectively collect Pezizales fungi and get spore prints. And the main takeaway here is that you want to place your cup fungus in something like a sterile petri dish. Not everyone has access to that. You could just use a clean plastic container, and you want to put your cup fungus in that container and put it close to the lid. You can use substrate, leaves, moss, or paper. Even in the field, you can bring something. And you leave that for a few hours so that the cup fungus is creating a spore print on the lid, on the plastic lid. You can even induce a simultaneous spore release. And here's a photo. If you've never done this, next time you find a cup fungus, give this a try because it's really fun. If you take a cup fungus and blow on it, you can make all the acini release their spores simultaneously. And it creates this kind of chimney effect where you get this cloud of spores coming out. And if you listen closely, you can even hear a hissing sound, which is pretty mind-boggling to think that that's all the microscopic cells simultaneously releasing 8 spores, and collectively that's creating a hissing sound that you can hear. So Discinaceae fungi, they are maybe sometimes difficult to get spore prints because they can mature very slowly. If you can collect, maybe come back in a few weeks and collect again if you don't get a spore print. But you can get spore prints from Discinaceae fungi, and it's really important to do so for microscopy on their spores. I have a little wish list here for people who are out looking for these things. All of the discoid Discinaceae, tribe Discineae specimens and fungi are undocumented, and we need more collections of them. A lot of these known unknowns I've been talking about, we might only have 1 or 2 representatives of them. So we need a lot more of these sequenced discoid Discinaceae species. Blancomyces from Alaska would be exceptional. We know that we have 2 undescribed species from Alaska, but all of our collections are from the 1950s and 60s. So something modern with good photographs would be excellent. More carefully documented Gyromitra specimens like Splendida and Vetinata would be really important. And of course, more Hypnotria, these truffles, since there's such high biodiversity in that group. And people are asking if I personally want specimens. Thank you for the offer. I can't take specimens right now. I don't have any place to store them. But hopefully in the future, I will be in a lab again where I can receive specimens. But I will, our last point here is hopefully you can sequence your collections. And there are some free sequencing services that you can use. Mycota Lab has multiple states where if you're in that state, you can send specimens for sequencing for free. Fundus, the Fungal Diversity Survey, has some projects and funding you can get access to. There's also the Ohio DNA Mushroom Lab that for some time was sequencing stuff for free. If you have a local a mushroom club, a lot of clubs are getting involved with DNA sequencing. So yeah, hopefully if you find Discinaceae, you can get them sequenced. If you sequence something, it gets propagated through a global network of researchers who are looking at this. If it's just photos, if it's just a specimen, it's interesting, but honestly, given the complexities of this group, it's difficult to fit it anywhere without sequence data. And here again is this, or here is this map from Mycota showing if you're in any of these states, you can send in specimens and you can get them sequenced for free. So check out MycoMap if you're in one of these states. This network is expanding every year. I don't know how they do it, to be honest. I don't know where they get the money from or the time or resources, but they are producing sequence data for specimens sent from these states. So take advantage of these services, and surely you can find novel things that could really expand our understanding of fungal biodiversity. And then Christian here is saying they're starting a fungarium at University of Alaska Fairbanks and would love to hold specimens. That sounds really great. With that, we're going to end with my conclusion. So in this talk, we looked at Lorchel taxonomy, ecology, and Gyromitrin distribution, informed by our phylogenomic research. I also talked about all these different Discineae genera, and we learned how we can identify them based on macromorphology and spore morphology. We talked about how there's at least 75 species in Discinaceae, of which 14 are known unknowns, either undescribed or we don't have the right name for them yet. And surely there's many more taxonomic novelties expected with increased sampling. That's because many areas of the world have no sequenced Discinaceae specimens. Perhaps with the same intensive sampling we're seeing from the Western United States through initiatives like the California Fungal Diversity Project, where they have actually people being paid to go out and collect mushrooms and everything's getting sequenced, that could be why we're getting so much novelty from the Western United States. And maybe it's actually not special. Maybe we would find this novelty anywhere if we had the same sampling effort as we're seeing from states like California and Washington and Oregon. And here's an example of a fungus, Gyromitra chirpoensis. This was described in 1972 from Costa Rica. It probably belongs to the Paragyromitra group. You know, there is nothing sequenced from Costa Rica. And it's really interesting. There's not a lot of reports at all of Discinaceae fungi down in like subtropics and tropical areas. But here is the species that has only ever been mentioned once in its initial publication. And there's a number of species like that. So I think with increased sampling, we're going to see the number of Discinaceae species grow a lot. At the same time, it's really a manageable number, and we've been doing a lot of work with this group resolving species concepts. So I'm hopeful that in my lifetime, we might have a really good understanding of the fascinating biodiversity present in the Lorchel family Discinaceae. So thank you everyone very much for your time and for listening to my talk. And with the remaining time, I can answer any more questions we might have.
Thank you, Dr. Dirks. That was awesome. I've been sitting here watching it and enjoying it. And I do think we might have a few more questions coming in. I see a lot of people on the chat thanking you and telling you how great this presentation was. So take a minute and read that and enjoy all the positive comments that people have. So thank you,
Yeah,
everyone, for sharing
thank you
those.
everyone.
We have recorded this session. It will be posted up on YouTube and on our webinar page probably within about 24 hours. So if you want to recommend it to somebody or see some of our older recordings, they are there for you to watch. But it looks like we're good. I think we should sign off now and say goodnight to everybody. And hopefully we'll see you again soon.
Yeah, I love that. Yeah, thanks everyone, and go look for some Discinaceae in if you're out here in Eastern North America, you know, March, April. I don't know how it's like out there in the West. It's always been confusing to me when you all get mushrooms, but soon enough for us. But yeah, thank you everyone and have a great night.
Good night. Good night, everyone.